Hello everyone. Firstly, I would like to thank the organized committee for inviting me to participate on the fourth edition of a global webinar on endocrinology and diabetes 2021. As you know, primary macronodular adrenal hyperplasia is considered a rare cause of Cushing syndrome, generally due to bilateral adrenal macronodules and often associated with a variable cortisol secretion and consequently cause a heterogeneous clinical presentation. PMAH was first described by Kirchner in 1964. In that time, PMAH was considered to be somewhere in between adenomas and carcinoma due to metabolites excreted that is more comparable with patients with adrenal carcinoma. However, up to now, no case had malignant behavior. PMAH has received different denominations over the decades according to new features identified. The prevalence of PMAH was thought to be less than 2% among all cases of endogenous Cushing syndrome. Due to more radiological image assess, patients with mild hypercortisolism has been identified, so the prevalence seems to be higher than previously thought. Related to molecular and genetic data, several molecular and cellular alterations of CMP pathway have been described in adrenal tumor, including PMAH. Activation of PKA pathway by GPCR was one of the first mechanisms described in PMAH. However, no genetic mechanism was able to explain these abnormal expressions. In 2001, our group observed for the first time the presence of ACTH immunostat in both adrenal glands and from patients with PMAH. In 2013, Luzet and co-author published in New England Journal of Medicine a robust paper showing that cortisol from PMAH is not truly ACTH independent, since the ectopic ACTH in clusters of adrenal cells stimulate in paracrine and nattocrine fashions of cortisol secretions. The authors showed that adrenal ACTH levels were higher in adrenal blood samples than in peripheral venous samples, consistent with a local production. The release of ACTH was stimulated by ligands of aberrant hormone receptor, so the PMAH hypercortisolism appeared to be ACTH adrenal dependent. In addition, more recently, they identified that ACTH was detected in a subpopulation that expressed gonadal marks. These observations suggest that ectopic ACTH may result from impaired embryogenesis, leading to abnormal maturation of adrenal gonadal primordium. Germline and somatic pathogenic variants of several genes involved in CMP pathway have been reported in PMAH. Nevertheless, a recent study using DNA fish analysis identified that somatic duplication of chromosome 19 in PMAH tissues without any duplication in germline DNA. However, these events occur only in a few cases. Polyzygotic mutation of GNAS causing McKinnon-Brett syndrome and neonatal patients can rarely present Cushing syndrome due to nodular adrenal hyperplasia. The same somatic pathogenic variant was described in adrenal nodes of PMAH, however, only in a few patients without any association with McKinnon-Brett syndrome. This mutation causes inhibiting of GTPase activity of G protein subunits and maintain constitutively activated the CMP-PKA pathway. MEN1 is a tumor suppressor gene involved in control cell cycle and proliferation. Knockout mice of MEN1 develop adrenal tumor or hyperplasia associated with corticosterone producing, so PMAH could be considered as one of the manifestations of MEN1. Recently, one study investigated the presence of variants of ARMCS in patients with MEN1, and two pathogenic variants associated with MEN1 were identified. The author suggests that the ARMCS has a role in modifying the phenotype of patients with MEN1 beyond its known role in macronodular adrenal hyperplasia. Overexpression of GPR have been demonstrated to promote adrenocortical tumorigenesis in mice model. In addition, GPR overexpression has also been associated with Cushing syndrome in PMAH. So we evaluated GPR expression in adrenal tissue from 3 patients with MEN1 syndrome in comparison with a human normal adrenal gland. The expression was 2.6-fold higher than normal adrenal gland. The tissue study did not present manual LOH, suggesting a potential role of GPR in development of adrenocortical hyperplasia associated with MAN1. HLRCC is an autosomic dominant disease characterized by cutaneous and uterine leiomyomas and aggressive kidney cancer, and also present adrenal hyperplasia in rare cases. Germline inactivation of pathogenic variants of the FH was described. Even with a low frequency, it could be considered a rare cause of PMAH. The incidence of adrenal incidentalomas in familial adenomatosis polyposis coli was reported at 7 to 13%, which was higher than in general population. However, PMAH was extremely rare. The activation of WNT beta-catenin pathway is involved in adrenal tumors, especially in adenomas and carcinoma. FAPI is caused by germline inactivation of APC gene, a tumor suppressor gene that inhibits beta-catenin signaling. The role of activation of beta-catenin as a driver of adrenocortical tumorigenesis has been clearly demonstrated in vitro and in vivo. Before 2013, all genetic defects describing PMAH explain only a few cases. As I told you, several observations pointed to a genetic origin of PMAH, as the report of a rare familial forms and the presence of bilateral adrenal nodules, although some can present asynchronous development. Using different approaches, integrated genomic study and the SNP-based genome-wide linkage analysis, followed by whole genome sequence from a large family with 96 members with PMAH, ARMCS gene was implicated in the pathogenesis of PMAH. The identification of ARMCS represents a great leap in knowledge than PMAH. During 20 years, only 13 patients were identified. Since the discovery of ARMCS, at least 45 families were described in 7 years. ARMCS belongs to the armadillo repeat family. It is located in chromosome 16 and present with 6 exons in the most common isoform and with 2 extremely conserved regions among species, the armadillo domain and BDP domain. In a functional study, ARMCS performed in adrenal cancer cell line. From Guillaume reported that the first time that the overexpression of ARMCS leads to cell death. Moreover, ARMCS silence decreased expression of ARMC2R MRI and steroidogenic enzymes and cortisol producer. We confirm the role of ARMCS in a more suitable biological model, PMAH primary and secondary cell cultures, as an important pro-apoptotic protein involving PMAH-related steroidogenesis. We reported for the first time the involvement of ARMCS in cell proliferation, cell cycle regulation. However, this effect needs to be further explored. A few years ago, it was reported that knockout mice of ARMCS represented with defects in development compromise immune stem and T cell differentiation. They also present with adrenal hyperplasia and increased corticosterone with aging, which is quite similar to macronodular adrenal hyperplasia in humans. In the same study, it was observed that ARMCS depends on the interaction with other proteins through the proteins-protein interactions. They observed that ARMCS causes neural tube defects. Here you can say small defect on mice. I looked for the presence of spina bifida in our patients, but the frequency of this find in primary macronodular adrenal families were similar to general population. Here you can just find that ARMCS is a part of a novel specific ubiquitin ligase A3, and its mutation is modifier of a neural tubule defect risk. CoL3 is a protein that participates in the UPS and mediates the regulation. The interaction of ARMCS and CoL2 has been observed by the different approaches such as mass spectrometry and double hybrid, but what is the role of ARMCS in interaction with CoL3? To answer this question, Cavalcanti for the first time evaluated these interactions by performing co-IP assay. The most important result of this study is ARMCS is a substrate of CoL3, and this interaction requires BTP domain. ARMCS plus CoL3 ubiquitination of ARMCS leading its degradation by UPS. ARMCS silence or overexpression alter cell cycle. This effect was blocked by Q3. Missense mutation in BTP domain of ARMCS are neither regulated by the Q3 UPS nor alter cell cycle. This new data suggests a novel mechanism of regulation of ARMCS, opening new perspective in understanding of the tumor suppression action of ARMCS and its role in PMA development. This is a model of biallelic inactivation of ARMCS to PMA development. In patients with germline mutation, secondary somatic event occurs over the time, leading to development of macronodules and hormone secretion. Most of the diagnoses are made at 50 decades when patients present over-Cushing syndrome or after the investigation of an adrenal incidentaloma or active investigation of relative index cases. There aren't hot spot regions on ARMCS. Few papers had analyzed the genotype-phenotype correlation, and it seems not to have a consensus. However, it looks like patients with ARMCS mutation present the largest adrenal glands and higher levels of cortisol secretions, but it needs to be further explored in a larger multi-center cohort. The next few slides I will show you some cases with PMA to demonstrate the clinical and biochemical heterogeneity of PMA. Here we show the hormonal data and the follow-up of two sisters with PMA and different somatic events of ARMCS. The first patient never developed adrenal insufficiency after adrenal sparing surgery. In addition, she recovered hypercortisolism after 2 years. The second patient presented adrenal insufficiency after surgery and maintained remission of Cushing syndrome in the follow-up of 7 years. According to different somatic mutation, we analyzed the sliced tissue of both patients, and the patients one present more compact cells, around 60% on adrenal tissue than her sister, 30 similar other patients. We showed this interesting data on end 2019. This patient referred for us with a severe Cushing syndrome and a low stat performance associated with a nonsense pathogenic germline variant at exon 3 of ARMCS. The patient deceased due to Cushing syndrome complication before surgery. You can see abnormal CT with an extraordinary value of Unidad's Housefield and also a high SUV on PET-CT. ARMCS expression is not limited to adrenal gland, and the different isoforms have different patterns of expression, and it is generally ubiquitously expressed, suggesting ARMCS may have a role in pathogenesis of the diseases involving other tissues beyond the adrenal gland. Since in 2005, PMA has been associated with meningioma, and the second hit was proved by Elbert in 2015. Interesting, we have been observing patients with PMA and meningioma and wild-type ARMCS. This is one of my work that is going on. Gastric inhibitory polypeptide receptor can be ectopically overexpressed in adenoma and PMA nodules and causing food-dependent Cushing syndrome. Interesting, no patients with food-dependent Cushing syndrome present ARMCS germline pathogenic variant. A new important genetic finding was discovered by two independent groups, one from France and the other one from Mail in Canada. Using pangenomic study, is identified the gene KDM1A as a tumor suppressor involved in almost cases of food-dependent Cushing syndrome. So, which scenarios should be suspected for PMA diagnosis? One scenario is when the patients present clinical features related to hypercortisolism, mild, cyclic, or over-Cushing syndrome, or you can observe a worse control of metabolic syndrome and classical signs and symptoms of Cushing syndrome. The other scenario for diagnosis of PMA is when you actively make investigation of relative index cases. In our family, we observe an incomplete penetrance of the disease in an old woman with germline mutation with normal cortisol after DST test without any adrenal nodules in a long-term follow-up. The number 29 in this genealogy is the oldest, and even with ARMCS5 variant germline, she remains normal. We can also investigate PMA when associated, as I told you, with genetic syndrome. The index case and risk relative associated with ARMCS5 germline variants. You can see the DST test is normal in no patient is that present at least one nodule and on adrenal gland. In addition, the majority of patients present low levels of DHES. They have followed this family since 2009. And they remained with a mild hypercortisolism and did not develop over Cushing syndrome, except for the one brother that came for us 2 months after the first index case. In addition, the patient with only one adrenal nodule up to now remain unilateral. This data reinforced the name primary macronodular adrenal cortical hyperplasia that covered all radiological and clinical presentation. We evaluated the frequency of variants in ARMCS5 adrenal in patients with unilateral and bilateral adrenal incidentalomas, all of them with mild hypercortisolism, and also in patients evaluated to Cushing syndrome associated with bilateral adrenal nodules. A multi-retrospective and prospective study compounded by 123 patients. And only one a family with adrenal incidentaloma was discovered. Here you can see the abdominal CT scan of patients with bilateral adrenal incidentaloma associated with ARMCS5 variant. We can also see the CT from her cyst and her niece. Both of them absolutely normal without metabolic syndrome and no features of Cushing syndrome. In addition, her cyst present acoustic meningioma. And here we can see the hormonal data of the familial PMA. The youngest with 31 nowadays maintain the normal values of cortisol after the DST test. The sister has mild value of cortisol after DST. We remain observing this family without any treatment. The reason they don't present any sign or symptoms of metabolic syndrome or osteoporosis, even with ARMCS5 variant, I don't know. When and how to treat this patient? It's a difficult and good question, mainly when you don't have a classical Cushing syndrome. A detailed follow-up of patients observed if there is a worsening of the metabolic disease or signs of a hypercortisolism, then you will be more secure in treating patients. However, you should keep in mind that even when the patients have a mild hypercortisolism secretion, they have a cardiovascular increase risk, and adrenalectomy has shown to improve mainly the blood pressure and control for these patients. For some ones, we can only wait and see. For other ones, we can use medication against GPR receptors, associated or not with heterogeneous inhibitors. Other ones you can indicate unilateral adrenalectomy with all or without medication. You can indicate adrenal sparing surgery and also indicate bilateral adrenalectomy as the worst possibility. According to our data from the investigation of aberrant hormone receptors in our families with PMA, we observe a lack of answer uniformity in the same patient and the same family. On the answer of protocol, we did not use any treatment against abnormal receptors. Therefore, we use this protocol for research only. Here we have indicated adrenal sparing surgery for these patients. That means total adrenalectomy of the large adrenal gland and partial adrenalectomy of contralateral adrenal gland. Initially, we use a functional imaging to determine which of them would be removed. However, nowadays we don't need to perform PET-CT FDG since the size of adrenal gland is correlated with the PET, implicating more cortisol production. This is the hormonal follow-up of the patients who undergoing adrenal sparing surgery. All patients develop periods of adrenal insufficiency and received replacement with a glucocorticoid. Some of them needed mineral corticoid replacement, and the majority of patients recovered the normal production of cortisol. Few patients needed glucocorticoid, and all patients was cured of a hypercortisolism with a normal DST test. Here we can see a patient with 70 years of age. She comes for us due to a mild hypercortisolism and central nervous system with meningioma. In addition, she presents a possible cortisol secretion and mild feature of Cushing syndrome, such as proximal myopathy. We discussed with her and her family about surgical procedure since she was worsening the diabetes control. The patient improved the diabetes control, hypertension, and the most important, she improved her cognition, and nowadays she comes alone to her appointment. To favor the independence to the elderly patients is really improving their quality of life. Here we have uncertain important points. PMAH is an underdiagnosed disease, so its real prevalence and incidence are unknown. ARMCS5 and KDM1A are the most important genes involved in PMAH, but they are not implicated in all cases. Aberrant hormone expression of receptors frequent in PMAH. However, more work is needed to understand the relation among ARMCS5, KDM1A, GPR expression, paracrine actions of ectopic ACT. Our understanding of genetic effects on phenotypes is far from complete, and the investigation of meningiomas, myelomas, and myelolipomas linked to KDM1A. When should we treat the patients? Only with Cushing syndrome? Have we treated with medication or with surgical procedure or with both? Which is the best surgical approach? Unilateral, bilateral, adrenal sparing surgery? So a collaborative multicenter study will need to improve the comprehension of these heterogeneous adrenal disorders in order to individualize the therapy for patients. Here, I would like to thank many thanks for all my fellows that give me a great pleasure to work together. And also our international colleagues, Jérôme Bertherat and André Lacroix, for the long travel together. And also a special thanks to Brazilian families that have mailing in the largest one. I am open for questions. Thank you very much for your attention.